Thanks a lot for the intro, Anne. And thanks all for coming here despite the nice weather outside. So I'm working for a Dutch biotech company, and I'm leading their lab that is using a technology which is called phage display. And this is a technology that is using the principles of evolution together with viruses to develop new medicines. And in the next 20 minutes, I hope to entertain you with some of the science that I'm really excited about. And just a disclaimer, I had to simplify a bit in this talk to make sure that it's accessible to all of you despite your diverse backgrounds. So before I dive into the details, I'd like to give you an overview of the process that is required to develop a new medicine. So it does start here, where you have to identify a target protein that is essential, for example, for a virus or a cancer cell. And ideally, this protein is something like an Achilles heel. So if you switch this protein off, the virus or the cancer cell dies. So if you identify this, then you start to screen huge libraries of molecules, and these can be contain thousands or millions of different compounds, and you try to find one molecule that can switch this protein off. If you succeed, then you move to this stage here where you study the in vitro biology of this protein. So what you try to do is you try to understand exactly how does this molecule bind to the target protein, how does it switch the protein off, but also, for example, if your molecule binds to other protein, which could later lead, for example, to side effects. And the two most probable outcomes are that either you have a compound that is promising, but it's way too weak, so then you have to go back here and screen another couple of thousands of similar compounds with the hope to find something better. Or the other most likely outcome is that something unexpected is happening on this level because there was something unknown on the biology, which then increases your understanding of the biology, but that probably means you have to restart this whole process. So if you find in the end something that is a very potent and promising molecule that could be a potential drug, then you have to test this molecule into an in vivo model in a mouse, for example, and that is not a nice thing, but that is required because on the next stage then we have to test these molecules in clinical trials in humans, and there are very strict regulations about this to make sure that the humans, or that the risk to harm humans is minimized. So if that's okay, then the clinical trials in humans can start, and they come in 3 phases. In the first phase, this compound is given to healthy volunteers to test if this compound is safe. If that's the case, then we move to the second phase, where this potential drug is now actually given to volunteers that are sick to assess if this drug can actually treat the disease. And if that's the case, the drug is then moved forward to the phase 3, where the drug is then given to a large number of volunteers to study if there are any long or side-term effects. And only if all of that is completed, then the drug is finally approved, so it can reach the market and the pharmacies. And I find that's a very interesting process because it covered nearly all fields of science, from chemistry to biology, medicine, biophysics, math, computer science. But this process has also some very interesting financial sides. So running through this whole process to develop a drug takes around 15 years, so there's a big chunk of a human life. The success chances are horrible. Only between 1 to 5 projects end up in a drug that makes it to the market. That makes this whole process also very expensive. The development of a drug costs between $1 to $5 billion. And the only reason why there's a whole industry that is dedicated to developing new drugs is that in some cases the sales for a drug can even exceed 10 billion. So this also means there's a strong connection between both health and economy. And of course, this raises a lot of social, ethical, and philosophical questions. For example, who's going to pay for these drugs in society? Should we only develop drugs for first world diseases like obesity, or also for malaria, for example, a disease for the third world? And should we, for example, should we consider aging as a disease and try to cure it? And I think that's all really good questions. Unfortunately, I don't have the answers to these because my expertise lies in these first 2 segments here, which span the first 2 to 3 years of these 15 years progress. And for me, that's the most exciting part, because on one side, this is where this whole story begins. but also because it allows you to play some kind of biological Sherlock Holmes and play molecular Lego, because what you try to do is to find a molecule that is kind of fitting to the cancer protein, for example. And the traditional way to do this is by exploring so-called small molecules. Here you can see an example. So this is a molecule that exists in nature in the bark of the willow tree, and for hundreds of years the tea of that bark has been used to treat pain. And in the last century, chemists then identified this compound in the bark and named it aspirin. And this compound can nowadays be synthesized, so you can just walk into a pharmacy or even a supermarket and spend €1, and then you can buy a package of aspirin, so you don't have to feel any more guilty that a whole forest of willow trees had to be chopped down just to treat your hangover. And well, that's a good case, but for a lot of diseases, there's no magic tree bark or magic mushroom available to be used or to be searched for a drug. And then the only way to find a drug is to find a small molecule. And that is actually a very, very challenging task. And the reason for this is that these molecules actually do look very simple, but there's actually such a high diversity of variants possible, and nobody knows the exact number, but the diversity is expected to be somewhere in the order of 10 to the power of 60 to 200. So this is such a large number that even if you would have all eternity and you are trying just to synthesize a single molecule of all of these different variants, then at some point you would probably run out of atoms, because the number of atoms in the universe is only expected to be 10 to the power of 80. So because of this reason, the search for a new drug has also been compared with the search of a needle in a haystack. And well, the only thing you can do is that you keep on searching and hope at some point you will be lucky to find something. So industry tackled this challenge by making this gigantic libraries or compound collections that can contain millions of compounds, and then they built like this dedicated screening center, which are huge complexes of buildings that are manned by very sophisticated robots that can roam around, and you have some robots that can take out these trays, and then these robots can pass on the trays to some other robots, and these robots can then pick an individual compound and then test if this compound binds to your cancer protein. So this is like a very, it's a brute force approach, because it allows you to test over 10,000 compounds per day, but building this type of infrastructure also costs hundreds of millions of euro. And despite all of this investment, actually your chance to find your needle in the haystack is still pretty low. And this forced then scientists to look for a solution elsewhere. And some scientists then found inspiration in nature, because nature developed this amazing problem-solving strategy, which is called evolution. And evolution allowed life on Earth to adapt, adjust, and evolve really from the deepest trenches in the ocean to the top of the mountains to survive asteroid impacts and the ice ages, and finally to create like the world that we are now living in. And if we humans manage at some point to really kind of break the world apart, then this might turn humanity into an evolutionary dead end. But I am pretty certain that some life will even survive us and then continue the success story of evolution. And one of the driving forces of evolution is natural selection. And I found this comic in the internet. And I don't think it needs any more explanations. But I was looking at this comic, and I remember that actually the Dutch are the tallest people in the world. And I started to wonder, what's actually hidden on top of the supermarket shelves that normal people cannot reach. So how does evolution work on a molecular level? So you all know that the blueprint of life is DNA, and DNA contains the building plans to build proteins. So this information to build a certain protein is copied from the DNA and another molecular format that is called RNA, and the RNA is then used as a template to build a protein. So mutations can occur in the DNA, and many of these mutations probably don't have any effect on the protein. But some of these mutations in the DNA might lead to changes in the protein, and some of these changes may lead to a long neck in giraffes, which is a positive effect in Africa, so natural selection will help to promote this mutation, but not, for example, in Europe, where apparently yummy leaves also grow on small trees. And this whole process is facilitated by proteins. So proteins are single molecules, and well, they're actually molecular machines, and they really surpass anything that we humans built so far. So they're only nanometer in size, but they're still able to do the job 1,000 times faster and to do less errors than any human machines. And it is not per chance that these proteins are so powerful machines, because they are in themselves the product of billions of years of evolution. And when people realized that these proteins are so powerful, they started to consider to use them for research and for drug discovery. And for this, they turned to phages. So phages are a type of virus, and they can come in all kind of different cool shapes. So they can have like this rod or worm-like shape, or my personal favorite is this one here that looks a bit like a space invader. And this is, of course, an artist impression, but the cool thing about these guys is that they actually really look like this. So this is an electron microscope picture, and you can see here the phages, and the phage sit on top of a bacterial cell, because that's what the phages do. Phages infect bacteria. So they are not harmful for humans, and they're not even considered as alternative to antibiotics. So what the phages do, they bind to a protein which is called the F peleus, which is presented on the outer membrane of the bacteria. And the bacteria need the F peleus for horizontal gene transfer. In other words, the bacteria needs it to have sex. And this actually then makes phages as a type of HIV for bacteria. And the phages do bind to this F peleus protein, and then they inject their DNA inside the bacterial cell, and then they hijack the molecular machines of the bacterial cell to make more copies of the phage DNA, RNA, and then to produce phage proteins. So in short, this turns the bacteria into a factory to produce more phages, and these phages are then secreted and can go and infect new bacteria. And that's quite a complex, that's a very complex process, but actually phages in themselves are very simple organisms. So they're basically just an empty vessel, and inside this empty vessel is a DNA of the phage, and the DNA is in the shape of this circle here, like a rubber band, but the rubber band is twisted to fit inside the phage. So the DNA of the phage is very simple. It only consists of around 10 genes, which is nothing compared to the complexity of us humans. We have around 25,000 genes, and this might make you feel now like we are the kings of creation, until you realize that this little guy, for example, here, that just lives in the canal outside of the building, has 30% more genes than we have. So we are maybe not that good. But nevertheless, phages are, from the genetical point, very easy to understand. And at some point, scientists learned how to hack these phages. So what they learned is if they take the DNA of the phage and they introduce a new piece of DNA at this position, and then they take the DNA, the phage DNA, and put it inside a bacterial cell, then the bacterial cell will start to produce a new phage, this one here. But in this case, it's different because this new type of phage has now a new protein displayed at its tip. And this new protein here was encoded by this new piece of DNA that you introduced here. And you can use this process to generate libraries of billions of different phages. And of these billions, each phage contains a unique protein at its tip. And this protein is encoded by the DNA that is inside the phage. And with this, you can now solve the needle in the haystack problem of drug discovery. And you're doing this by taking your cancer protein, you immobilize this on a solid support, then you take your solution containing your billions of different phages, and the vast majority of your phages will display proteins that do not match the cancer protein. But there will be a few candidates that display a protein that matches the cancer protein, and they will bind to this protein. That's what you can see here with the red phage. And this allows you then to retain the phages that do bind to the cancer protein, but to wash away all the phages that do not bind to the cancer protein. Then you can recover this phage, and you can use it to infect new bacteria. And these bacteria will then produce more phages of the same type. This gives you now a new population of phages that is enriched in binder. And you can repeat the cycle for multiple rounds until you end up with a population of phages that is highly enriched in binder. At this moment, you still don't know what actually this protein is that can bind to your cancer protein. But at this point, then, you can read out the DNA that is inside of the phage, and this will tell you then what type of protein was displayed on the tip of the phage. So the technology is called phage display, and it's a part of the technologies for directed evolution, because what this technology allows you to do is to select variants from a gene pool for a certain purpose. And it's a very powerful technology because it allows you to screen billions of different variants in less than a month. You don't need any expensive machinery for this, and you can basically carry around your library just in an Eppendorf tube. So it's not a huge building you have to carry around. And this technology is now quite widely used in drug discovery. What you can see here is Humira. This is the first drug that was actually developed using phage display. You can see it's much bigger than the aspirin that I showed you before. And this drug is also quite important because it was very successful from the medical point of view. It's used to treat certain types of autoimmune diseases. It was also very successful from the financial point of view. At some point, the sales of this drug exceeded $10 billion per year. So the phage display and directed evolution technologies are now widely used in science, and this was recognized with last year's Nobel Prize in chemistry, where these 3 laureates were awarded for harnessing the power of evolution. One of these people, George Smith, was a scientist that discovered the principle of phage display, while Sir Gregory Winter was a scientist who realized that you can use this then to solve your needle in the haystack problem. And he was also the person that founded the company that developed Humira, so probably doesn't have any financial issues during retirement. As I mentioned, I work for a biotech company called PepScan, and we use this technology for drug discovery, but you can also use this technology for the development of diagnostics. So if you have one example where we collaborate with another company, and together we developed an assay for precision medicine. So what we can do with this is that we take a drop of blood from a patient that is treated with one of these antibody drugs like Humira, and then we found with our platform a molecule that binds to this antibody, and we can use that to quantify how much of this drug is in the patient. And that is an important piece of information, because if you have here the concentration of the drug, then you will get different effects. If the concentration is too low, you don't have a therapeutic effect, so the drug is useless. But if you have a too high concentration, then you will get toxic side effects. And if you will take your package of aspirin and read the instructions, there are 2 different instructions. If you're a child, you should take half a tablet. If you're an adult, you should take a full tablet. But obviously, there's a lot of differences between all of us here, and people realize now that it's important to adjust the dosing of these drugs on a patient-specific level. So this allows you then to increase both the safety and the efficacy of these drugs, but also to reduce the cost of these drugs. And this, I come to my final slide, which has nothing to do with what I told you so far. But I recently read a piece of research from the Erasmus University where they studied the online behavior of adults. And they realized from all the vast information that is available online, what apparently most people are really interested in is to look at cute animal puppies. So I did some research myself, and I found like this picture, but I also find 99 other pictures that were equally nice, and I had to save all of them on my hard disk. And I do hope that in 2 months you will still remember this talk, and if not for the Nobel Prize level science, then maybe for this cute animal picture.